Hi Nana. So today the topic is immunoglobulins. So what are the immunoglobulins and why they are important in our immunity? What is their shape and on which basis they are classified? All the things we have to discuss in this class. First of all, I have to tell you that what is immunoglobulin? Immunoglobulin. If you see the word, it has two parts. Immunoglobulin. Immunoglobulin. Globulins are the proteins. The proteins which will give immunity to an individual. Once again I am repeating you Nana. The proteins which will give immunity to the individuals. Such proteins are called as the immunoglobulins. These are the proteins. Immunoglobulins are the proteins. They will give the immunity. They will give the immunity. To whom they will give the immunity? The host which will prepare these immunoglobulins. They will give protection to that host only, right? So in simple way, if you have to describe about immunoglobulins, you just call them as your army. You know what the work of army? They will protect our country, right? So in the same sense, these immunoglobulins are army of our body. They will protect our body against the pathogens or foreign substances which will enter our body and causes so many diseases, right? So today we are going to discuss about these immunoglobulins. And next thing, which cells will produce these immunoglobulins? So the B lymphocytes and plasma cells. B lymphocytes and plasma cells. These two types of cells produces the immunoglobulins. Okay, and the shape, if you see the shape, they are in Y shape. The immunoglobulin, a typical immunoglobulin will looks like in the shape of Y. So this is the very short introduction of immunoglobulin. But when you go into the depth, there are so many interesting things. It means how they protect the host body, right? So we will go into the deep of the subject. Okay Nana, I already told you that the immunoglobulins are Y shaped. Let us see what is the detailed shape of an immunoglobulin. If you see the immunoglobulin, it has two pairs of protein chains or polypeptide chains. We will call it as polypeptide chains, or you may also call them as what are these amino acid chains also, because the proteins are made up of amino acid sequences, right? So these are the two pairs of polypeptide chains which are combined and formed the immunoglobulin. So see here, this is one pair. These are very short, right? Hence these are called as the short chains, and these are very heavy or long, right? These are called as the heavy chains or long chains. Okay, heavy chains. These two are the heavy chains and these two are the light chains. And one more important thing, these two light chains are very short in size, and these two heavy chains are very long in size. And one more important thing, these chains are interlinked with one another. So for the strong bonding, the interlinking is very important between these two pairs of polypeptide chains. So what they will do, they will form disulfide bonds. They will form disulfide bonds between the two heavy chains and between the light chain and heavy chain. See this carefully Nana. The disulfide bond are made in between two heavy chains, and they are also formed between one heavy chain and one light chain, and they also formed in between the same chain also. Why means for the strong bonding, they formed disulfide bonds among the chains and in between the chains. It is just called as the intra-chain disulfide bonds and inter-chain disulfide bonds. Inter-chain means the chain which was formed between the two different chains. It was the ఇంటర్చైన్ టూ చైన్స్కి మధ్యలో బాండ్స్ ఫామ్ అయితే వాటిని ఇంటర్చైన్ డైసల్ఫైడ్ బాండ్స్ అన్నాము. అండ్ if the disulfide bond was formed among the same chain, it is called as the intrachain disulfide bonds. So intrachain disulfide bonds form అవుతాయి, interchain disulfide bonds కూడా ఫామ్ అవుతాయి. ఓకేనా నాన్న? తర్వాత these immunoglobulins have a very high molecular weight. Okay, and the next thing, it has N terminal and C terminal. For all the four chains, upper one is the N terminal and lower one is the C terminal. This is the C terminal, this is the C terminal, this is the N terminal, and this is the N terminal, and this is the N terminal. Okay na nana? And the next one more important thing, if you see, this is the called as the variable part, and remaining part it is called as the constant part. This is the constant chain and this is the variable. Enduku? Why it is called as variable and why it is called as the constant means, I already told you, these are the polypeptide chains, means made up of amino acids, right? So the amino acid sequence of these four polypeptide chains was changing time to time depending upon the antigen shape. మరి మొత్తం చేంజ్ అయిపోతుందా అండి? సో వన్స్ అగైన్ ఐఎమ్ టెలింగ్ యూ, ఇది మొత్తం ఒక యాంటీబాడీ స్ట్రక్చర్ అనుకున్నట్లయితే ఓన్లీ హాఫ్ ఆఫ్ ద పార్ట్ ఈస్ చేంజింగ్ ఇట్స్ షేప్ అండ్ రిమైనింగ్ హాఫ్ ఈస్ కాన్స్టెంట్. రైట్ నానా? హాఫ్ ఆఫ్ ద పార్ట్ ఈస్ చేంజింగ్ ఇట్స్ షేప్ అకార్డింగ్ టు ద యాంటీజెన్ షేప్ and half is constant. So the part of the antibody, okay, the part of the antibody which is changing its amino acid sequence according to the antigen, just like this nana. If the antigen was in this shape, the antibody must be produced in this shape. If the antibody is this shape, sorry, if the antigen is like this shape, the antibody must produce like in this shape. Means the antibody is produced depending upon the shape of antigen. Why? Because for antigen antibody reactions, right? So in the same sense, according to the shape of antigen, the antibody shape also changing. How it will be changed means the amino acid sequence must be changed. So depending upon these amino acid sequence changes, the antibody is divided into two parts. The upper part is the variable region and the lower part or half of the part, remaining one is the constant one and upper one is the variable region. Okay nana? And next thing is, so these two are the light chains and these two are the heavy chains, right? So depending upon the amino acid sequence of these light chains, these are two types. Light chains are two types. Light chains are two types. Light chains are two types. ఏమేంటి అండి అంటే కప్పా chain and lambda. Sorry. Lambda chain. So depending upon the amino acid sequence, the light chain are two types, kappa chains and lambda chains, and most of the light chains of antibodies are kappa model only, right? And very rare antibodies or immunoglobulins appears in the lambda chain. And okay ma'am, it was about the light chains. Then what about the heavy chain? Right, for heavy chain also, depending upon the amino acid sequence of a heavy chain, they are divided into 5 types. G, A, M, D, E. This is the short form for these 5 types of immunoglobulins: Ig, G, immunoglobulins, gamma immunoglobulins, and alpha immunoglobulins, and mu immunoglobulins, and delta immunoglobulins, and epsilon immunoglobulins. G for gamma, A for alpha, M for mu, and D for delta, and E for epsilon. Gamma immunoglobulins, alpha immunoglobulins, mu immunoglobulins, delta immunoglobulins, and epsilon immunoglobulins. IgG, IgA, IgM, IgD, and IgG. One more thing, these immunoglobulins are shown with the IgG. These are immunoglobulin word represented with IG, right? So here one more thing I have to tell you, in antibodies or immunoglobulins, the two heavy chains must be single type only and the two light chains must be a single type. This is the already fixed by the nature or fixed by our body. As I told you earlier, kappa chains and lambda chains, if you see one light chain is kappa and one light chain is lambda, what it will be happen? The bonding will be very quite difficult, right? So there is a fixed nature, that is the two light chains must be a single type and two heavy chains must be a single type. సో ఇక్కడ ఉన్న రెండు హెవీ చైన్స్ ఒకే టైప్ అయి ఉంటాయి, అయితే G type or A or M or D or E, and at the same time the two light chains must be a single type, either kappa type or lambda type. Okay na? So the difference may be between the two heavy and two light, but there is no difference between the 2L and 2H. అంటే రెండు లైట్ చైన్స్ మధ్య డిఫరెన్స్ ఉండదు, రెండు హెచ్ చైన్స్ మధ్య కూడా డిఫరెన్స్ ఉండదు. మరి దేని మధ్య ఉంటుందండి అంటే రెండు హెచ్ చైన్స్ గామా అయి ఉండొచ్చు, రెండు లైట్ చైన్స్ కప్పా అయి ఉండొచ్చు, లేదా రెండు లైట్ చైన్స్ ల్యాండా అయి ఉండొచ్చు, రెండు హెచ్ చైన్స్ ఐదిట్లో ఏ టైప్ అయినా అయి ఉండొచ్చు. ఓకేనా నాన్న? సో ఐ హోప్ యూ అండర్స్టూడ్. థ్యాంక్ యూ.